Good morning. In this video, we are going to discuss about buffer systems present in our body which are needed for regulating the acid-base balance and for maintaining the normal pH. The normal blood pH is between 7.38 to 7.42 and the intracellular pH is maintained somewhere between 7, around 7. So this normal range, whenever it changes even slightly also, it can affect the charge on the protein. If the charge is affected, it in turn can affect the function of various important proteins and enzymes present in our body. So it is very important to maintain the pH within this narrow normal range. But it is not an easy task. As a result of metabolism, loads and loads of acids are continuously produced in our system. For example, from proteins, especially during the catabolism of sulfur-containing amino acids, we get sulfuric acid. From lipids, especially during starvation, we get keto acids. From carbohydrates, during anaerobic conditions, we get lactic acid. And from the catabolism of nucleic acid, we also get phosphoric acid. In addition to all this, in TCA cycle, which is the final common oxidative pathway of all these nutrients, we will be getting carbon dioxide. When this carbon dioxide dissolves in water, it will be producing carbonic acid. So all these acids will try to decrease the blood pH very drastically. But to prevent this, our body, our system is equipped with three major defense mechanisms, which are nothing but the first line of defense is buffers, second line of defense is lungs, and the third line is kidneys. Okay, so in this, the first line, the buffer system, we will be discussing in detail in this video. So regarding this buffer, you should be knowing five important things. The first one, first you should know what is a buffer, next what it is made up of, then how it acts, then how effectively it can act, and finally a brief description about the various buffers present in our body. First, let me start with the definition for buffers. Buffers are compounds which resist the changes in pH, but it can resist only when a small amount of acid or alkali is added. If a large amount is added, it cannot withstand. Okay, so which means buffers are defined as compounds which resist the change in pH only when a small amount of an acid or alkali is added. This buffer is usually made up of either a weak acid and its conjugate base or a weak base and its conjugate acid. The best example for it is carbonic acid along with sodium bicarbonate. Here carbonic acid is a weak acid whereas sodium bicarbonate is its conjugate base. By taking this as an example, I will explain how the buffer system works. Suppose when a strong alkali like sodium hydroxide is added to our system, what will happen means the acid part of our, when a base is added, the acid part of our buffer will come for the rescue. So that means the carbonic acid, it will dissociate to form H plus and HCO3 minus. In this, the H plus will bind to the OH minus to form water, whereas the bicarbonate HCO3 minus will bind with sodium to form sodium bicarbonate. Okay, so what had happened? The sodium hydroxide, the strong alkali added, has been converted into a weak base and water so that the pH will not change much. On the other hand, when a strong acid like HCl is added, our base part of the buffer will come for the rescue. So the base part means sodium bicarbonate. So they will dissociate to form sodium and HCO3 minus. In this, sodium will react with chloride ions to form sodium chloride, whereas bicarbonate will react with the H plus ions to form carbonic acid. So the strong acid added is converted to a neutral salt and a weak acid so that pH will not be changed much. This is how the buffer system works in our body. When acid comes, base will come for the rescue, and when a base comes, acid will come for the rescue. Okay, so how effectively it can buffer, the buffer can act means that is dependent on two important factors. The buffering capacity of any buffer depends on two important factors. One is the pKa value of the acid and the second one is base by acid ratio. In this, pKa value is nothing but the pH at which the acid will be half dissociated. That means when the base by acid concentration will be almost same. Okay, so normally the pKa of the buffer, it should be very close to the pH of the working system. That means plus or minus 1 to the pH of the working system. For example, if a buffer should act good in an extracellular pH, what is the normal extracellular pH? It is somewhere around 7.4. So if the pKa of the buffer is between 8.4 to 9.4, then it can act as an effective buffer. Okay, if the pKa value is plus or minus 1 to the pH of the medium, the system where it should work, then it will act as an effective buffer. Okay, next coming to the second component, base by acid ratio. Ideally, the base concentration and acid concentration should be equal. Then only both it can act effectively against both the acids and alkali. But in our body, just now in the initial slides we saw that in our body it is mainly the acids that are produced. So to counterattack that, we need large quantities of bases. Okay, so which we call it as alkali reserve. So we need a base excess or alkali reserve. Only then it can act as a good buffer in our human body. Okay, so these are the two factors. Either pKa should be close to the blood pH, or otherwise the base concentration should be little high than compared to the acid concentration, so that it can act as an excellent buffer within our human body. Okay, so that's it about the buffering capacity. Next, coming to the last thing, so the few buffers which are having a good buffering capacity inside our human body are bicarbonate buffer, phosphate buffer, protein buffer, and finally hemoglobin buffer. Now I will give a brief description about each one of this buffer system. First, let me start with bicarbonate buffer. So in this bicarbonate buffer, like any buffer, I said it is made up of a weak acid and its conjugate base. So here its composition is carbonic acid and sodium bicarbonate. Okay, so carbonic acid is a weak acid and sodium bicarbonate is its conjugate base. So this bicarbonate buffer made up of these two components, it acts as an excellent extracellular buffer. It is one of the best extracellular buffer. So why it is considered as an excellent extracellular buffer means, though its pKa value is not in the pH of the ECF. Okay, so I said pKa value should be plus or minus 1 of the pH. What is the pH of the blood or the extracellular compartment? It is 7.4. So ideally the pKa of the buffer should be between 6.4 to 8.4. But here its pKa is just 6.1. Though the pKa value is not optimal, the base by acid concentration is very high. The alkali reserve is very important for acting in our body. Okay, so that alkali reserve or base excess is excellent with the bicarbonate buffer. That is why it is considered as a very good extracellular buffer. Okay, so in addition to this advantage, excess alkali reserve, there is also one more advantage with this bicarbonate buffer, because both the components, carbonic acid and sodium bicarbonate, they are under the physiological control. Their levels can be regulated either by the lung or kidney accordingly. Okay, so because of these advantages, bicarbonate buffer is considered as a good extracellular buffer. Next, coming to the second type, phosphate buffer. So here the phosphate buffer, the acid part is NaH2PO4. The one with extra hydrogen is called as acid part, whereas the one with less hydrogen, Na2HPO4, it is called as base part. Okay, so this is the weak acid and that is a conjugate base. So like in bicarbonate, I said it is a good extracellular buffer, whereas this phosphate buffer is a good intracellular buffer. Okay, so why it is a good intracellular buffer means it pKa values exactly same that of intracellular pH. I said the intracellular pH is around 7. Okay, the pKa value is almost close to 7. That is why it acts as a very good intracellular buffer. Okay, apart from that, even the pKa ratio, the acid by base by acid ratio, it is also 4:1, which is relatively good. Not as good as bicarbonate, but relatively good. But here the plus point for phosphate buffer is its pKa value is very close to the intracellular pH. Okay, apart from intracellular buffer, it can also act as a good urinary buffer. So most of the acids excreted by the kidney will be removed only as a phosphate buffer. Okay, so that's it about phosphate buffer. Bicarbonate buffer is a extracellular buffer. Phosphate buffer is a intracellular as well as urinary buffer. Next, coming to the third one, protein buffer. So this protein buffer can act both in intracellular compartment and also in extracellular compartment, but predominantly within the tissue cells. Okay, in the extracellular compartment, the major protein that acts as a buffer is albumin, because that is the major plasma protein. So in this albumin or any other protein, the buffering capacity is determined by the concentration of histidine residues. The amount of histidine residues is the one which decides the buffering capacity of any protein, because it is only the imidazole group of histidine. The pKa of the imidazole group of histidine is close to the human body's pH. Whereas all the other amino acids, their pKa will be far away from the human pH. Only the histidine group is having the closest to pH. That's why the amount of histidine residues present in the protein will determine the buffering capacity. Okay, so that's it about protein buffer. Next, among these proteins, we have hemoglobin, which has a very important role in acid-base regulation. So what does that role means? We know that all the cells in our body, they take up oxygen and they will release carbon dioxide. If all these carbon dioxide are poured into the blood, what will happen? It will dissolve in the aqueous blood and it can produce carbonic acid, and that carbonic acid can lead to acidosis. In order to prevent this, what is happening means almost 75% of the carbon dioxide released from the tissues is immediately taken up by the RBCs. Okay, so once it enters into the RBC, it will dissolve in the water. Even within the RBC, we have water. Okay, so it will dissolve in the water to form carbonic acid. It is catalyzed by the enzyme carbonic anhydrase. Okay, so now this carbonic acid will dissociate immediately to form H plus and HCO3 minus. In this, the H plus will bind to the histidine residue of hemoglobin and it will be buffered so that the pH within the RBC will not be altered. Okay, whereas the bicarbonate produced will be thrown out of the RBC. Okay, into the circulation it will be thrown out. For every bicarbonate thrown out, to maintain the electrical neutrality, one chloride will be entering into the RBC. This is called as chloride shift. Okay, so this is how hemoglobin helps in buffering the H plus ions which are produced from the carbonic acid. Okay, so now this RBC which has buffered the H plus ions will go to the lungs. In the lungs, what will happen? The partial pressure of oxygen is very high. So what will happen means the partial pressure of oxygen will displace the H plus ion and they will release the hemoglobin H plus from the hemoglobin. Now this released H plus ions will bind with the bicarbonate ions present in the RBC to form carbonic acid. As the bicarbonate ions present in the RBC is used up, more and more bicarbonate from outside will be entering into the cell. Now again, for every bicarbonate entering, one chloride will be thrown out of the cell, which is called as reverse chloride shift. Okay, now coming back to the equation. So once the carbonic acid is formed, so this carbonic acid will dissociate to form carbon dioxide and water, and that carbon dioxide will be expired out, will be exhaled out through the lungs. This is how the carbon dioxide which is formed in the tissue is taken to the lungs by the hemoglobin without affecting the pH of the blood. Okay, so this is function of hemoglobin is called as isohydric transport of carbon dioxide. Okay, carbon dioxide was produced in peripheral tissues. Okay, from that, if it directly goes in blood to the lungs, what will happen? It may dissolve and it may form carbonic acid and it can cause acidosis. Okay, but instead of that, if it enters into the RBC, it can be effectively buffered by hemoglobin and it can be transported to the lungs without changing the pH. That is called as isohydric transport of carbon dioxide. Okay, so that's it about the various buffer systems in our body. Okay, so to summarize, we have 4 major buffers in our body. In that, the excellent extracellular buffer is bicarbonate. The excellent intracellular and urinary buffer is phosphate buffer. Hemoglobin helps in isohydric transport of carbon dioxide, whereas in protein, the histidine residues helps in buffering action. Okay, so protein is actually a minor buffer. Bicarbonate is an excellent extracellular buffer, whereas in the intracellular compartment, it is mainly the phosphate that acts as a good buffer. Okay, thank you.